Thank you very much for the very kind introduction and for the invitation. It's really an honor to talk here. And so I'm going to discuss the neural network mechanisms of flexible, robust, and efficient cognitive motor control. But before diving into this very topic, I'd like to just take a step back and to briefly discuss the challenges and hopes of brain and cognitive sciences. So what we all would like to do is to try to understand how the brain generates behavior. But the brain is this complex, partially observable system where it's hard to distinguish principle versus detail. And behavior is adaptive, flexible, and robust in complex environments. And that makes it challenging to understand or emulate with artificial intelligence. And so in this context, I think that theory can be very powerful because it can allow us to select a parsimonious set of constraints on the architecture, on the dynamics from the brain. And these constraints are hypotheses for principles that can help us explain how the brain generates behavior. And we can also generate prediction about what the activity of the brain should look like in conditions that you haven't used to derive the theory in order to test the theory. And along the way, there can be rich interactions with the field of artificial intelligence. And so we've said we'd like to try to understand behavior, and I've been interested in motor behavior specifically. So I'm just going to take a few minutes to just describe main characteristics of biological movements. Maybe obviously it's a dynamic, analog, and smooth. And it's not just that it makes pretty picture, but smoothness is actually linked to energy efficiency. So for instance, when humans move their hands in between two positions and reach, the speed of the hand is going to undergo this bell curve over time. And it's not just that this bell curve is smooth. You can show that it's very close to minimizing jerk, but you can also show that this also corresponds to the most metabolically efficient solution. So dynamic, analog, and smooth, that's one characteristic. And another characteristic is that it combines internally generated dynamics with sensory-driven corrections. And the most obvious demonstration of this presence of internally generated dynamics is a little bit extreme. It comes from patients who have lost their touch sensations. They have no somatosensation. You can have the patient come in the lab and ask the patient to close their eyes and to perform actually very fine movements. For instance, here the patient is going to touch all of their fingers in a sequence with their thumb. And what's interesting is that they're going to be able to do this even with what should be basically no sensory input here for several seconds, which is a very long time scale relative to the single neuron time scale. And it's only many, many seconds later that you start to have a drift. But of course, this is an extreme case. Most movements are going to have to integrate sensory-driven corrections, for instance, if you're interacting with an object. Perhaps if you have a very quick, reliable sensory input, then you can think of the network that's going to implement that as a simple feed-forward map between the sensory input and the motor command. But if the sensory input is delayed or very noisy, then you can think of this sensory input as a correction that sits on top of internally generated dynamics, which rely on a recurrent network, and which is consistent with what we saw in the patient. And those internally generated dynamics are what we are going to focus on today. So I've been starting to introduce a few networks here. What do these networks look in the brain? So there's an evolutionary preserved architecture for the flexible control of actions. And in mammals, this involves notably the motor cortex, which is thought to generate motor output because mammals have evolved this pretty direct pathway from motor cortex to very peripheral motor structures. And motor cortex interact with higher order cortical regions, which monitor sensory input as well as contextual information. And in the end, they can produce abstract action selection signals. And these selection signals are going to be broadcast to other brain areas, including subcortical regions such as the basal ganglia and thalamus, which has this very specific loop architecture and connects back to motor cortex. And in this talk, the questions that we're going to ask is, how can we interface the slow abstract action selection signals that are on the right with the motocortical dynamics that is on the left that produces the continuous movement during flexible motor sequencing? And we're going to be very interested in the role of the architecture of this circuit, the thalamocortical circuit on the left. So in order to pose the problem very clearly, I want to pose it in terms of behavior. So really what we were interested in is our ability to learn individual motor motifs, which you can think of as different dance moves. And what that means from a motor control standpoint is our brain learns to generate a temporal pattern, like this squiggle here, that is going to be driving our muscle during a particular movement. And learning each dance move can take a long time. But once you have learned a library of those motifs, when the music comes, you can flexibly assemble them in arbitrary sequences in response to abstract, potentially internally generated commands. In addition, you can learn new motor motifs without interference and include them anywhere in the sequence. So this was what we were trying to understand. And in order to understand how the brain does it, I was interested in accounting for the architecture of the brain regions that are involved in this. And I've already presented to you these basal ganglia brain region that is going to transmit some action selection signals to motor cortex through thalamus. And thalamus until recently was thought to be more of a relay back to cortex. But actually, there's also this projection from motor cortex to thalamus that forms these loops. And these loops have been shown experimentally to be important for cortical dynamics. And they're also going to be at the core of our model. And we're going to try to understand what they do in the model. So these are architectural constraints, but I was also interested in the electrophysiology, the electrophysiological signatures at different parts of the circuit. So first, in motor cortex, the activity undergoes two phases. First, a phase of preparation, where, for instance, if the monkey is going to make two different reaches, the activity is going to converge towards a particular static activity pattern that is going to be identifying the identity of the reach. And then during execution, the activity undergoes some oscillations. And these oscillations have the very interesting property that a weighted sum of them is going to be very good at fitting the activity of the muscles, the EMG. And so the idea is that motor cortex acts as a dynamical machine that generates motor motifs. And there's an ensemble of evidence for this, and that's something that we are going to introduce in our model. We are also going to account for known electrophysiological signatures at the other side of the circuit in the output nuclei of the basal ganglia, where there are sequence control signals. Specifically, some neurons are going to have increased activity during a particular motif, and others are going to have more phasic activity at the borders between motifs. And we're going to see that this might be linked to preparation. And because the basal ganglia signals that are sent to thalamus are inhibitory and they can be strong and they could turn off the neurons in thalamus, we're going to ask, what is the role of these resulting thalamus activity switches in configuring the cortical dynamics for motor preparation and execution? And to ask that question, my approach has been to develop a model that was complex enough such that it could reproduce the main activity features that we discussed and do the task, but simple enough that we could analyze it. So it consists of a recurrent network, motor cortex, which has a readout that is going to be producing the temporal patterns that drive the muscles. And cortex interacts in a bidirectional fashion with thalamic units. And there is no excitatory recurrence in between thalamic units, which is a very preserved architectural feature. And that's really what's important in our model too. And these thalamic units receive these inhibitory inputs from the basal ganglia signals. These inhibitory signals from the basal ganglia. And the activity in thalamus that we modeled, we described it with a rectified linear function. Specifically, the thalamic units that receive basal ganglia signals are going to be assumed to be rectified. And the thalamic units such as the ones that are left free to interact with cortex are going to be assumed to stay in the linear regime. And the thalamic activity Z is also going to be assumed to be faster than cortex because of the absence of recurrent excitation in thalamus, which involves that the neurons in thalamus are governed by their intrinsic timescales, while cortical neural populations, which drive movement, have slower timescales. So what we get is the thalamic activity Z is expressed as a product of a selection matrix S, which is diagonal and just has ones to index the neurons such as the ones that are left free to interact with cortex and zeros else. And it has to multiply the input received by thalamic units such as Z1, which is the product between the cortical weights R and the corticothalamic weights JCT. So that's for thalamus. And then for cortex, we are going to consider linearized dynamics. That means that the cortical weights are going to be governed by these differential equations that involves recurrent interaction through the cortical-cortical connectivity matrix JCC, as well as the input from the thalamic activity Z through the thalamocortical weights JTC. And what we get when we replace Z here is that in the dynamics, we're going to have linear bipart dynamics, which means that the connectivity matrix is going to be fixed as long as the identity of the thalamic units that are being inhibited doesn't change. But the connectivity matrix will also have a perturbative part here, which is the addition of a matrix that involves the thalamocortical and corticothalamic weights of the units such as Z1 that are left free to interact with cortex. So what that means with this switching linear dynamics is that we can use linear dynamical tools for analysis as long as the thalamic activity pattern doesn't change. But we add the fact that we have a nonlinearity in the dynamics, which we are going to see can be useful to increase computational power of the model. Interestingly, it also makes the model consistent with observations that within movement motor cortical dynamics is well captured by a low-dimensional linear dynamical system and actually better than some alternative nonlinear models. So having described the dynamics, we can also describe other constraints of the model, which is the location of learning. So we're going to assume that all the red synapses are fixed, and we're only going to try to learn through the blue synapses. So why would we do that? It's because if basal ganglia now selects non-overlapping thalamic populations to drive different motifs, we're going to be able to learn different motifs using completely separate thalamocortical loops, so separate synapses. And that could have the power to avoid interference. So that's good, but what's not clear is whether this works at all and how well does it work. So to try to understand how well does it work, we should first try to understand how the isolated motor cortex works. We remove thalamus for now. So motor cortex this model is just a dynamical system. Its activity will undergo a trajectory over time. And we can also fully describe its activity in this case by an ensemble of basis functions, so the eigenmodes. And these basis functions, they are just the projection of the activity along specific directions in the activity space, which are the basis axis or eigenvectors. And these basis functions are basis functions because they have a fixed shape. They are going to be exponentially modulated sine waves. And they are interesting because the output is just the sum of these basis functions. So if we understand the properties of these basis functions, we understand something about the type of outputs that this network can produce. And here we can be completely clear with what the effect of fixing the connectivity is, which we need to avoid interference when learning different motifs. So when we fix the connectivity, we fix the basis axis, and we also fix the timescales of the basis functions, the exponential timescales and the oscillatory timescales. And they are going to leave these timescales in a restricted portion of the space of possible timescales, depending on the statistics of this connectivity. So this leaves us with only 1 degree of freedom to control the activity of the network, and that's the initial rates. What these initial rates control is the initial amplitude and phase of the basis functions. That means that if we change these initial rates, we can change these amplitude and phase, and we can produce different output variants. But we can only work with the basis functions that we have, which have the same interesting timescales. So what happens now if there is a mismatch between the timescales that are in the desired output and the available timescales? Well, in that case, when we adjust the initial rate the best we can, we're still going to have a mismatch between the produced output and the desired output. And so we're going to have an inaccurate output with plausible rates, not fine-tuned, not extremely large. So this is a limitation, and that's where we can try to understand what the thalamocortical loops can do to help us. And we're going to consider the minimal case of a single thalamocortical loop with thalamocortical connectivity U and corticothalamic connectivity V. And the reason why we do that is that if we can show that this loop can do a lot, then we could do even more with small loops. So what that means is that the connectivity is a sum between the fixed cortico-cortical connectivity. Again, this is fixed to avoid interference between motifs in this case. And this has n-squared synapses, so a lot of synapses. And we're trying to change it, to control it, with just the product between U and V, which is a rank 1 matrix here, which we're going to be able to learn. But that's only 2n synapses. So that's why it might be surprising if we can do a lot with it. So what can we do with it? So we're going to consider in sequence the 2 properties of the dynamics, which are the timescales of the basis functions and their axes, their directions. So first, the timescales. I'm just reminding you here in this schematic what the timescales of the isolated cortex looked like. And what you can show that's possible to do is to give yourself desired timescales that are outside of the ensemble of timescales of the isolated cortex. And what you can show is that you can just adjust or learn one connectivity here, the connectivity U, to make sure that the total circuit, so cortex plus loop, is going to include the timescales that you wanted. So we have the blue dots here that are just falling on the crosses. What we did is we changed the shape of these basis functions. That's something that has been done in the literature before. In control theories, there is also an ensemble of articles, notably by Omri Barak. But the specificity of what we did there is that we found and focused on a particular closed form equation for this connectivity U, which first can be shown to favor stable dynamics. It makes sure that the timescales of the perturbed cortex that we don't choose are pushed away from the real line indirectly. And the second thing is that this equation is very transparent for when it's going to be easy to change timescales and when it's going to be hard. So this equation is here, and it involves the basis axis, the eigenvectors of the isolated fixed motor cortex, the red matrix R here, the vector V that right now is random, we didn't touch it, and the important part, which is this matrix P. And we're going to have to take the pseudo-inverse of P, and P is has elements, the inverse between the desired timescales here, the crosses, and the original timescales here. So what we can see is that if we want to put many, many desired timescales away from the ensemble of original timescales, then we are going to have several rows of P's that are going to be correlated, and it's going to become more and more numerically difficult to invert. And so we're going to have some feasible cases where we can put, quite a lot of desired timescales away from the original timescales. But if we have many more timescales we want to put outside, or if they are further away from the original timescales, then there is too much correlation in the rows of P, it fails numerically, and biologically what that would correspond to is too large and or too fine-tuned weights. But this case here that's feasible is still very interesting because we were able to control many different timescales, which is basically much better than what we were having with the sum of the isolated motor cortex or the loop itself, which only has one real eigenvalue. So this case is interesting, and what that tells us is that we can adjust many timescales of the dynamics just with learning one part of this connectivity. But now, what about the basis axes? So they are going to change when we add the loop. So how do they change? So that's a case where we can actually we can have a closed form equation for these basis axes. And this equation is shown here. And again, it's going to involve the basis axis or eigenvectors of the isolated model cortex, model cortex in red. So the matrix R and L. The vector V that right now is random. And again, this term that is the difference between the timescales of the perturbed cortex and the original timescales. And we can see again that if we put several of these perturbed cortex eigenvalues much further away from the original ones, we're going to create correlations between these basis axes. And so these correlations are going to create what is called a non-normal matrix, which is capable of non-normal amplification, which can overlap with the readout. So what does it mean concretely? What it means concretely is that if we were able to exactly adjust the initial rate, then the desired output is going to match now our output because we have the right timescales in the circuit. But if we put just even a tiny little bit of noise in the initial rates, here just 1%, this noise can get amplified and we can get inaccuracies in the output. So we can quantify this in terms of readout error and also the error in every neuron's activity. For this perturbed matrix here with a random connectivity V, or from match matrices with the same timescales, but basis axes that come from a random matrix, which have limited amount of non-normality, or orthogonal basis axes that are really, they have no non-normality by definition. We can see that the error is much increased. So this is not great for our task. Luckily, we still have something we can do. We can learn this other connectivity V, and we will learn it to try to reduce the readout error. And we're going to see what happens. And what happens is that we're going to be able to get the readout error down very much, at times even close to what you would have gotten with an orthogonal matrix. Importantly, however, you're not going to be able in general to have all the error in all possible directions down. And that's because we only adjusted n parameters. So we can't make the matrix orthogonal. And how much remaining error in all possible directions remains really depend on how much you've been reshaping the timescales of the circuit. And so the important part of what we did is that we were able to orthogonalize the directions of noise amplification with respect to the readout. And what that allows us to do is to robustly read out the desired basis functions, such that now, if we adjust the initial rate to adjust the amplitude and phase, even if it's not perfect, we're going to have an accurate output. So what the loop did here is it acted as a module that can peripherally control the dynamics of the large cortex. And what it did is to increase the expressivity that we would have had compared to what we would have had if we didn't have the loop. And we did it with using very few learned parameters. Okay, so this was the role of thalamocortical interactions during execution. But what about motor preparation and trigger? So I told you at the beginning that during delayed reaches, there are these two phases of preparation and execution in motor cortex. And what we can relate it to is that we've seen that in order to produce the right activity pattern, in our case, we need to adjust the initial rates in order to set the amplitude and phase of the basis functions. And so the idea is that preparation can be the ensemble of events that are going to find and set the initial rate. And then the trigger is the time when these initial rates are now released in this network that is going to drive the muscles, such that its dynamics are going to execute the movement. And when we were working on this project, it was starting to be clear that preparation can be fast. It looks the same in a queue-initiated context where you have a long time to prepare, and in a quasi-automatic context. And the way you can see that is that this is the same neuron, and the darkness of the color indicates the direction. And you can see that the rates are identical for identical directions in the two contexts here. So preparation looks the same. But if we're trying to do preparation in a trivial way with an isolated model cortex, and what that really means is that we want the rates here to settle to a particular desired pattern. In that case, the trivial solution would be to just give an external input that's specific to the pattern you want the rates to converge to, and to just let it go to steady state, to final state. However, if you have dynamics in motor cortex that are good to control slow movements, then you're going to have convergence towards the right patterns that's going to be basically equally slow. So that's not ideal, and that led us to think that maybe thalamus could also help us here. And this idea was also spearheaded by the observation in the basic ganglia that there are these neurons that are active at transitions. And what these neurons could do is to inhibit the thalamic motif production units that we talked about before, and to leave a thalamic preparatory module free to interact with cortex. And what we were able to show is that in our article, we can optimize it for fast decay dynamics, regardless of what the dynamics of motor cortex is going to be next. And that's related to, but a little bit different from another great article by the group of Guillaume and Carles, if you're interested. So, but in this case, what really we focused on is, a few thalamic loops that, and the loops, so the black loops here are optimized in such a way that you go fast to the steady state, whatever the dynamics that you're going to experience next. And concretely, what that means is that when we add these loops here, we are able to shift the timescales away from the real line, which makes, again, the decay fast. And what concretely that means for preparation is that now if we want to prepare motif 2 and we give an input that's specific to motif 2, now the cortical rates are going to quickly go to the ideal pattern to start the execution of motif 2. And then we explain how this thalamic preparatory module can help us to prepare and trigger the network fast. So now we can see how the circuit is going to behave during sequences when we put together both preparation and execution. So let's say we want to learn 3 different motor outputs, 3 different motifs through these different thalamocortical loops, ZA, ZB, ZC. What we have seen is that if the motor cortex is correctly prepared for the preparation of motif A, when we trigger motif A, we just let the motor cortex free to interact with ZA, and then it produces the motif A. And if we want to now prepare and trigger motif B, so we first need to let this special thalamic preparatory / trigger network free to interact with cortex. And what this is going to do is combine with this input specific for motif B, the activity in motor cortex is going to converge towards the right activity pattern to start motif B. And then when we leave cortex free to interact with ZB, we produce motif B, and so on and so forth for the preparation and trigger execution of motif C. And so we can see that we can produce different sequences by just changing the order of the basal ganglia inhibitory pattern. And that means that we were able to do what we wanted to do at the beginning, which is to improvise new smooth motif sequences. And that's because we did not have any synapses here that was tuned to a particular transition. Instead, the synapses are tuned to separate motifs, or they are shared across all motifs and not trained for a specific motif. So that's nice. We're able to do what we wanted to do with this network, but can we say something about how efficient it is? And in order to start answering this question, it's interesting, I think, to compare it to the simplest networks that could exist and do the task, which would be a linear network. If we're trying to produce different motifs that require different timescales with linear dynamics, then you're going to need different subnetworks for these different motifs. So this is, for instance, the subnetwork for motif B. And if we're counting the number of units in this network, then it's going to be first determined by the number of units in each subnetwork, which is going to scale as a number of basis functions per motifs. It is going to then be multiplied by the number of subnetworks, which is a number of motifs. So we have this project here for this basal linear network. So what about the model that I presented, which has this switching linear dynamics? So the switch is a nonlinearity that gives this network the capacity in principle to do better. And the question is whether this capacity is realized, and it's only going to be realized if we have learned the synapses in a smart way. So were we able to achieve that? The answer is yes, and it's yes because with a single loop, we control many basis functions and therefore a complex readout. And because of this, in these networks, the number of units is just going to be the fixed number of units in cortex, which scales as the number of basis functions per motif. And as many thalamic units as we want to learn different dynamics with different timescales in order to produce many different motifs. And so we have this sum here, and this sum here is going to be smaller than the product there for long and complex sequences. So that suggests that this architecture on the right is well suited for producing these long and complex sequences. So that's an interesting model with constraint switching multilinear dynamics, which we built, keeping in mind that this is consistent with what was being seen in motor cortex. But a natural question then is to ask, can it do more experimental predictions? And since we started to develop that, has there been some validations? Indeed, actually, there have been such results that are consistent with the model recently. First, it's been now confirmed that thalamus is involved during the execution of fan movements, that the cortical thalamic pathways is involved in the learning and execution of movement, and that there are larger thalamocortical couplings during preparation and or trigger as opposed to during execution, which is also consistent with our model, because during execution, we only need to control the readout, whereas during preparation, we need to control the whole circuit. And in addition, consistently with our model, it's now being confirmed that when monkeys produce smooth sequences, individual elements of the sequence, individual reaches, are actually going to be separately prepared. That's also a feature that was present in our model. And a third point is that there starts to be analysis that suggests that very different movement can arise from different cortical dynamics. But really, it would actually be nice to test that further. And that's actually something that I have just started to get into, along with Adam Hartman, Stefan Lenke, and Ila Feet, in a task that uses sequences of varied arm movements. And we want to try to see, using simultaneous recordings in motor cortex and thalamus, whether there are switches in thalamic activities that precede switches in cortical dynamics during trigger and execution, and whether there are cortical dynamics during motor execution of different movements that could be compatible with Laurent control. And if we can get there by recording while the mice learn new movements, we'll see if we can get to the question of whether learning without interference can involve new move-specific thalamic units. That's nice in terms of where we stand and in terms of trying to move forward to further build on the model and revise it if needed. But this model also was built with some biological constraints in mind that are not always necessarily functional. And so a natural question that theories can have is whether some of the conclusions also hold in more general dynamics networks, which don't have a priori biological constraint. So the type of networks that I'm talking about here are networks which have continuous nonlinearity. So we don't decide when we switch or not. The nonlinearity can act at all points. We have a tanh nonlinearity here. And we focused on continuous time recurrent neural networks because we want to generate these analog smooth motor outputs. And the design principles that we're going to have here are just going to be basically stemming from the function. And the functions that we want to have is we want to avoid interference between motifs. And there are different ways of doing this, and I focused on different architectures that can achieve this by segregating the parameters that are involved in learning different motifs. And these parameters here, for instance, is the simplest example, are just an input to a fixed network. And these inputs are going to be learned by gradient descent, state-of-the-art algorithms such as ARM. And we can actually train single motifs in that way, and they can actually be very accurate, and we can train them in a noise robust regime by starting from random initial condition. And we can train easy ones, such as these smooth ones, or harder ones that have these discontinuities. And you can see that this doesn't seem to be involving any of the preparation or triggering mechanisms that I described before. But an interesting question is, you can train single motif, but can you transfer to motif sequencing? And I want to mention why you might want to be able to do this transfer. First, it can be because of time or memory constraints. So you want to avoid having to train on an increasing set of transitions. But really, I think that the most important thing is that in the real world, being able to just transfer zero shot to performing a sequence can be a safety issue. You want to be able to land in new circumstances, whatever other movement you've done before. So can we do this? So one way that in machine learning you can try to do this is to use randomization to try to favor generalization. So what that would mean is that we would want to have an approximate match between the random initial conditions that we're using during training and the initialization of motifs that are going to be experienced during sequencing, which are the end of previous motifs. And if we consider the type of networks that we looked at, we had an idea that a Gaussian function wouldn't be a bad prior for the random initialization, and you can play with its standard deviation to more or less match the random initializations that you're using and the end of motif activity patterns that you observe at the end of training. Of course, these patterns, these end of motif patterns here, they really depend on training. So it will look a little bit different each time, but you can get an idea with different iterations of what they look like, and you can more or less match this random initialization to these end of motif patterns. And then you can see how well that does. And what we are going to find is that there are actually relatively large errors that are going to be observed. And here, different colors indicate that this particular motif has been preceded by different other motifs. So if it was preceded by, say, a blue motif, it was very bad here. So that's a problem, and I've seen that these issues are pretty general. They're seen with different architectures, different motifs, network size. Even if you make the random initialization slightly larger than the end of motifs, and with or without weight regularization. So how do we fix that? An idea could be that maybe we can introduce a motor preparation / trigger module in these networks. That would be similar to the one we had in the switching linear networks. And the way we did that is, again, we used this idea of loops, which correspond to a perturbation of the weights. And this weight perturbation we trained in order to force the isolated recurrent network to go very fast to steady state. And then we fixed these preparatory weights, and we train the motif-specific parameters, which involve an input along with these preparatory loops for a little bit of time. And then either you keep the same input in the simplest type of architecture I showed you before, or you could also have a loop that is trained to produce the motif in a way that's similar to the thalamocortical model I presented to you. And so what we want to now compare is this architecture, which has this prep/trigger network, and which doesn't have any new parameters that's tuned to a particular motif, which is important. And we want to compare it to the more vanilla networks, which are closer to previous works that don't have this preparation. And as a reminder, with the latter one, we were doing okay with random initialization, but not so good during transitions. And now, if we introduce a preparatory module, not only do we transition well, but we also have a global improvement in performance. That really seems to be because we train better. And so what we can do is to quantify this with many different motifs, and we can see that with preparation, we do much better. This is not architecture dependent. And therefore, that suggests a general function for this motor preparation / trigger, which can go beyond evolutionary idiosyncrasies and suggests that this really is not a random thing that the brain implements, but it may have like a general function. So conclusions quickly. The first conclusion that I would like to make is really more technical. What was interesting to me is that we were able to come up together with the mathematical analysis of recurrent neural network, which combines a biologically constrained architecture and tuned synapses. And at least it wasn't obvious to me at the beginning how we were able to do that. But by leveraging switching linear dynamics and insights that can be gained with rank 1 perturbation of these dynamics, that was able to be achieved. The second point is about the conclusions that we actually made. That come from the fact that we were able to show that the thalamocortical architecture is well suited for preparation and execution of varied motor motifs. And that relied on leveraging shared cortical circuitry across motifs, both the recurrent network and the preparatory network. And also from using frugally motif-specific synapses. And these were containing loops that corresponding to low-rank perturbations that can powerfully adjust the dynamics. And we're also able to avoid interference when learning different motifs, because these loops were separate for different motifs. And the last point that I was able to show is that even though these insights were developed for a relatively simplified model, if we now combine recurrent neural network training with some adjusted insights for these types of modular dynamics, we can actually probe how general these insights can be. And in our case, we're able to give a normative account of motor preparation for sequence improvisation. And so in the future, I'm going to keep on focusing on motor control with questions of motor coordination and synergies, questions of sensory motor integration, and questions of network architecture for premotor circuits and planning. And with this, I'm going to thank you very much for keeping with me for this very long time. I'm going to thank my advisors, my collaborators, and my colleagues, notably Sean and Larry, who have been Sean Escol and Larry Abbott, who have been my advisors for the projects that I presented today. And Ila is supporting me Ila Fit is supporting me for pursuing the follow-up projects. And I would like to thank the funding, the Simons Foundation Transition to Independence Awards that supports me as I'm trying to transition. And I'm trying to transition within 1 to 2 years. So if you're interested in the research program and think that I could be a good fit, let me know. And also let me know if you should be interested in discussing this and in potentially working together. And we're going to have with as the people, Jonathan Michaels, Kaya Hardcastle, and Nama Hap as a cosine workshop on motor sequences. So you can also see us here and learn more about the biology there. Thank you very much.